Is insulin really the reason why we're all getting fat? Let's talk about it. I'll see you inside. So, initially, I actually wanted to film an episode about diabetes and a kind of paradoxical effect that occurs in diabetics. However, to really understand that episode, I think I should probably do an episode explaining what insulin is and what it actually does. Because I realize that not everyone is actually all that familiar with insulin. Additionally, there's a lot of differing information about insulin. Some people will tell you that insulin is the reason that everyone gets fat, and that the only reason body fat accumulation occurs is because insulin. Simply, there are some people who advocate for extremely low carb diets whose answer to every problem in the world is insulin. With that being said, we have professional bodybuilders who inject insulin in order to improve their physique. So it's obvious that insulin itself may not actually be a problem, but maybe having too much insulin is the problem. And to that statement, I would actually agree. I think it all comes down to your insulin sensitivity. You see, your body is not going to create and produce a hormone that's going to have a negative impact. With that being said, if we force our body to overproduce a hormone, that itself will have a negative impact. And the body is extremely elegant, and it will try to adapt to an overproduction of hormones. So even though that you've put the body in a situation where it's overproducing a hormone, the body has adaptive mechanisms to try and make everything normal again. And we will get into this soon, but first I think we should understand what insulin is. So as I said, insulin is a hormone, and it's made and secreted by the pancreas. Insulin is secreted when blood glucose levels begin to increase. This is what occurs after a meal. For the most part, the more carbohydrates you eat in a meal, the higher the increase in insulin will be. And the reason for that is because the more carbohydrates you ingest, the higher your blood glucose levels will go. This is why people who are afraid of the effects of insulin advocate for a low carbohydrate diet so that your blood glucose doesn't increase and there is no need for insulin production. And now you can kind of think of the function of insulin as a door opener. Insulin doesn't actually take macronutrients and put them into the cell. Rather, it allows the cell to express different channels for uptaking those nutrients. And now to be less abstract about this, cells have what's referred to as an insulin receptor. And the insulin receptor is located on the heart, the brain, the muscle tissue, and the fat cells, and other tissues inside of the body. And now, insulin will bind to the receptor, but it will not actually enter the cell. You see, the receptor sits on the outside of the cell, and connected on the inside of the cell is what is referred to as a G protein. And when insulin binds to the receptor, this G protein is actually released from the membrane and goes on to do a lot of different things inside the cell, which we will touch on in further detail later. However, we first need to talk about the production of insulin. As I mentioned, insulin is secreted in the pancreas. It's secreted by a specific cell called the beta cell of the pancreas in the islets of Langerhans. Insulin is actually stored inside of the cell in a little vesicle, and unless blood glucose levels go up a substantial degree, insulin kind of just sits in these cells. And now this next point is very important. On the beta cell, there's what's referred to as a GLUT2 transporter. Simply, this is a transporter of glucose that does not require insulin, which is very important because if the cells that produced insulin needed insulin to function, we would have a very faulty system. So since these cells have GLUT2 receptors, these receptors always sit on the membrane of the cell and allow glucose to enter the cell. Within the beta cell, glucose is metabolized into ATP. Conveniently, also on the beta cell is a potassium channel with an ATP binding spot, which means that the ATP we produce from glucose can actually bind to this potassium channel. And the result is that the potassium channel changes shape such that it stops letting potassium outside of the cell. And now this kind of explains why insulin increases when blood glucose increases. If there's more blood glucose, more glucose will enter the cell and more ATP will be produced. The more ATP that's produced, the more ATP will bind to these potassium channels and shut them off. So since I'm talking about them, these potassium channels must be somewhat important. So when the potassium channels are unable to let potassium out of the cell, the voltage of the cell will increase because potassium has a positive charge and it's accumulating inside of the cell. And conveniently, in addition to the potassium channel on the membrane, beta cells have what's referred to as a voltage-gated calcium channel. Simply, this calcium channel remains closed unless the voltage increases to a specific level. And potassium building up inside the cell will raise the voltage enough to allow the calcium channel to open. Therefore, the concentration of calcium inside the cell will begin to increase. And now calcium is very important, but before we get into calcium, we need to understand what synaptoproteins are. Simply, these are small proteins located on both the membrane of the cell and the membrane of that little vesicle we spoke about that contains insulin. What calcium is able to do is allow these two proteins to connect with one another and allow that vesicle to fuse to the membrane of the cell and release insulin into the bloodstream. Alright, now we can finally get into the functions of insulin now that it's in the bloodstream. So many people refer to insulin as a storage hormone, which is true. What insulin does is it binds to that receptor we spoke about and causes that G protein cascade. These G proteins have a series of reactions that are oftentimes different in different cells. However, the end result is actually transcription of the DNA that is located inside of the cell. And the things that are transcribed typically are little channels or receptors that allow macronutrients and micronutrients and water inside of the cell. So let's take a look at the muscle. Simply, insulin is the signal for a cell to express the glucose receptor on the membrane of the cell. And now I mentioned the glucose receptor called the GLUT2 receptor earlier. The GLUT2 receptor is not on muscle cells. Muscle cells actually have a GLUT4 receptor. The GLUT4 receptors are not actually expressed on the membrane of the cell without insulin. Rather, they're dormant inside of the cell. Insulin signals the GLUT4 protein to be fused to the membrane of the cell, which will allow glucose to enter the cell. So, insulin will bind to a specific receptor on the membrane of the muscle cell, and the result is an activation of what's referred to as the PI3K AKT pathway, which is very complex, and I can try to explain it to you without getting anything incorrect. And this is a very complex pathway, but what is very important to remember is that AKT is the end molecule. What AKT does is it binds to a specific protein on the GLUT4 receptor called AS160. This protein is actually inhibiting that GLUT4 receptor from fusing to the membrane. But through that PI3K AKT pathway I mentioned earlier, AKT gets phosphorylated, which simply means activated. And the AKT will phosphorylate this protein and inhibit the protein from working. So we have inhibited the inhibitor of the GLUT4 receptor. Try to say that 10 times fast. So the GLUT4 receptor can now bind to the membrane of the cell and begin to uptake the glucose. And now, with that being said, in order for the excess glucose to actually be stored, we must turn it into glycogen. However, there's a molecule inside of the cell called GSK3 that inhibits glycogen synthesis when insulin is not present. However, insulin is a signal to store glucose inside of the cell. And that AKT molecule that was produced earlier also inhibits this GSK3 molecule that is inhibiting glycogen synthesis. So once again, we have inhibited the inhibitor, and now we can store glucose as glycogen inside of the cell. And now the way that this is structured is pretty important. Simply, we don't just create glycogen when there is no reason to create glycogen. So technically, insulin is the storage hormone because it's insulin that causes the cascade to occur that allows glycogen synthesis to happen. Now, the other function of insulin is to increase what's called mTORC. And this is entirely too complex to even explain in words. But this transcription factor directly stimulates protein synthesis in the nucleus by transcribing specific portions of the DNA that code for cellular growth. So to put that in normal terms, insulin is the signal to increase cell growth through putting on muscle. It also is the signal to store glycogen because it is allowing more glucose to enter the cell. So if you think about it, the reason that bodybuilders would inject insulin is to increase the amount of muscle they are able to build because it allows for more muscle protein synthesis to be signaled inside of the cell. Additionally, some bodybuilders even go so far as to take insulin before a workout. And the reason they would decide to do this is because the insulin will allow glucose into the cell at a higher capacity. So, when they're exercising a muscle, there's more glucose present to turn into energy to keep going and keep pushing through a workout. Alright, the next cell type I want to talk about is the liver cell. So, insulin binds to the same insulin receptor on the liver. The glucose receptor on the liver is also a GLUT2 receptor that is always expressed on the membrane of the liver cells. So regardless of insulin, glucose can always enter the liver at a rate dependent on the blood glucose levels. Thus, high blood glucose means that there will be elevated glucose inside the liver. And insulin binds to that same pathway we talked about, the PI3K AKT pathway. However, it doesn't trigger a GLUT4 receptor to the membrane of the liver, but it does serve the same glycogen synthesis effect as in the muscle tissue. So what insulin will do is increase the amount of glycogen we store. And now on fat cells, we have the same PI3K AKT pathway and the same mTORC pathway. However, the DNA that's encoded codes for enzymes that are required for what's called lipogenesis, which is simply the creation of triglycerides within the cell from glucose and amino acids. Thus, the result is increased fat cell size due to increased triglycerides. It also increases the uptake of fatty acids that are eventually turned into triglycerides as well. And now, insulin obviously has a multitude of effects depending on the specific tissue it's acting upon. It seems that in the brain, insulin has the same effect as it does on the muscle tissue and increases glucose uptake, which the brain then simply just uses as energy to fuel all of the cognitive functions that we need. So, insulin is very complex and it works differently pretty much everywhere. It aids in the storage of fat, carbohydrates, proteins, and water. And while I did my best to portray some of the roles of insulin, there are so many other roles, such as water, as we talked about. Some cells have what's referred to as an insulin-dependent sodium channel. So when insulin goes up, more sodium can enter the cell, and along follows water. Simply, there's just so many things that insulin does that it's hard to pinpoint just one mechanism of action. However, I hope this episode helped kind of understand what insulin is and what it does, and why sometimes it's beneficial and sometimes it's not. And why we can't just say insulin bad and we should all lower our insulin as much as possible. In our next episode, I hope I'll be able to explain to you a little paradox that occurs when we have very high insulin levels all the time. So I hope to see you there, and thanks for listening.